Good morning, colleagues. Thank you so much for taking time to just give the background. I think we've been running. Thank you so much, Dr. Lachayo. Like Dr. Lachayo said, that we've been running a series of talks on prenatal diagnosis and also the prenatal diagnostic test. We opened up the meeting today because the topic we want to discuss is a topic which I think is very important for every practicing obstetrician to be cognizant of, because there has been a lot of developments in the field of prenatal diagnostic tests. So we thought it is prudent that we update each other and so that we can counsel our patients in the right way. So to just give a quick recap, because I'm aware that not everyone has managed to attend our previous meetings, so that everything makes sense, or I put everything in the context as I present, I'll just make a very quick recap of the prenatal diagnosis test procedures. This is just probably, people don't need to know this checklist. This is for people who work in the fetal medicine units. But we talked about amniocentesis. It is important to know that when we take the fluid surrounding the baby, the sample contains fibroblasts, which are coming from the baby's skin. These are the cells that are in the amniotic fluid. It is important to take note of that. Right, then it is important again to realize that the argument between early versus late amniocentesis was settled with the CEMAT study, which was, and in the CEMAT study, this is the summary of the findings. That if you try to do it earlier, you are going to meet a lot of technical difficulties. We are also going to meet a high rate of karyotype culture failure. You are going to meet a high rate of pregnancy loss. You are going to meet a lot of club foot and also transverse limb defect due to the transaction of the amniotic sac, because the amnion and the chorion are not yet fused before 15 weeks. So, and there is also going to be increased risk of leakage of amniotic fluid. So amniocentesis should not be done before 15 weeks. Before 15 weeks, CVS should be done. Of course, this is probably for those working in fetal medicine, and I'm just doing a quick recap of last week's presentation so that those who are joining us today can make sense of what we are talking of. Then we also highlighted that even if an amniocentesis is done properly, there are certain challenges that you may meet. We may have culture failure, which may occur in 0.1%. We may also have mosaicism. Mosaicism is the existence of two karyotypes in an individual. In individual's colonies, when they are analyzed, we may discover that there is a monosome X and also an euploid karyotype in the same individuals. If this is present in all the colonies, then it's true mosaicism. But if it is in one, then the rest don't have, then this will become pseudo-mosaicism, which doesn't matter really. Then, of course, we may contaminate with maternal cells if we don't discard the first 2 meals or the first meal after putting in the needle. Don't know why my slides are refusing to move. Right. So we mentioned some of the complications, which we said they are very rare. Fetal loss, I'll show you the latest evidence on what we know about amniocentesis and also CVS, that the fetal loss is very low, is not as what is reported in all the books that we probably, some of us used. Then the fetal trauma is very rare, because right now we do it under ultrasound scan guidance, and the leakage of amniotic fluids, which occurs in about 2%, self-resolves, and we might never know of it because it's leaking into the abdomen. So, but it self-resolves, and there are no reported cases where women became oligoadrenic because they've leaked amniotic fluid post the procedure. Then amnionitis is reported again less than 0.1%, which is the risk of infection. Then the risk of transmission from these conditions, HIV, hepatitis B and C, and CAV is very rare in the presence of normal viral suppression. So it is important, on our checklist, you have seen that we always want to know if the woman has had this kind of test done before we do the diagnostic procedure. Then rhesus alloimmunization again is very rare if the anti-D is given after the procedure. But if it is not given after the procedure, there are other certain sort of precautions we take, like avoiding going through the placenta. Chorionic villi sampling, the most important image which I want us to recall is that the sample contains cytotrophoblasts and mesenchyme cells. Cytotrophoblasts already have a high mitotic index. They are already multiplying in numbers, while mesenchyme are in this G0 phase of the cell cycle. They are resting cells. So that means the sample from CVS allows direct examination because we already have cells that do not need culture. That is the cytotrophoblasts. The only challenge is that these cytotrophoblasts are filled with division errors because they are multiplying a lot, so that a direct examination should always be followed by proper culture of the mesenchyme. Okay, the indications, I think I'll skip through this because this is a recap of what we covered. Okay, chondrosynthesis, I'll also skip that. Then let me come to the evidence. The procedure-related risk has been evaluated in this systematic review and meta-analysis by Professor Koleka there. What we now know and what we tell women whenever we want to do the procedure is that the procedure is very safe because it is based from this analysis that looked at the weighted pooled risks of miscarriage following this procedure. Out of the 324 losses that occurred in 42,700 women who want amniocentesis, and those 207 out of the 8,819 who went we had CVS. So they noted that just the absolute loss in terms of the percentage was 0.81 for amniocentesis, and for CVS it was 2.18. However, they discounted for the what you call the background risk in a population, because there is always a background risk of miscarriage, which you should take cognizance of. And they noted that for the women who did not undergo any procedure, the background risk was 0.67 for amniocentesis, and for CVS it was 1.79. So the conclusion is that when we do the weighted pooled procedure-related risk for amniocentesis and CVS, it was 0.11 and 0.22. To say the miscarriage, which is occurring directly as a result of the procedure, is this low. It is very low. It's not the 1 and 2% which the books describe, but it is very low. And this is what we tell women before we do the procedure, that look, the procedure of miscarriage is very low. Of course, we are going to analyze our data. We are still doing them. We have not yet done 100. When we do 100, we also analyze our data so that we can tell you what we are doing from our own experience. But the procedure-related risk from amniocentesis and CVS is very, very low. Of course, expertise is very important when interpreting these figures. Right, so I'm going to go into today's talk, which is focusing on prenatal diagnostic tests. Prenatal diagnostic tests have undergone a lot of evolution in the last few years, probably because of this slide which I want to put here, which represent more than 100,000 cases done by the Fetal Medicine Foundation, where they were noting that if a baby comes, let's just use this last row to illustrate the point. So if we take a baby who has surely an abnormal knockout translucency of 6.5, which is way, way above the 99th centile, you would only find chromosomal defects when you do the conventional karyotype in about 64.5%. So that means the other babies, which are 35.5%, will be reported as having a normal karyotype. However, if you look at the alive and well rate, you can see a clear discrepancy that 20-something% of the babies who are actually reported as normal, they don't go on to be alive and well. Then this therefore means that surely there is something which this conventional karyotype is missing. Someone cannot just be sick. Someone cannot just die at birth or have an abnormality without there being an abnormality at the level of the gene. Somebody cannot just have a deranged neurodevelopmental status while the genes are absolutely normal. So these were the questions that people began to pose and say, is this conventional karyotype good enough? Why is it that even those who are reported as normal, they go on to have a poor outcome, and 19% of those with a normal karyotype, as you can see here, they go on to die, yet the karyotype has been reported as normal. So this therefore means that conventional karyotype is not good enough in diagnosing all the abnormalities at the level of the genes. So that means those who present with, and especially those who present with a fetal abnormality, it is not good enough to just offer conventional karyotype, because even if it comes back as normal, it does not necessarily translate to a normal baby. So babies with an abnormal knockout translucency above the 99 centile, they deserve to be offered more than the conventional karyotype. They need to be offered more, as I will continue my presentation. But to understand what I'm going to present, I will probably take you through some biology, some cell biology a little bit, so that what I'm trying to present makes sense. Then karyotype was compared with this method called chromosome microarray. I will describe it in my presentation. And they looked at the clinical utility to say, if you meet a baby with a fetal defect, where the karyotype has been reported as normal, or what percentage of babies that if you do a microarray, I will describe what a microarray is later on, it will come back as normal. So they noted that it was in about 6.5% of the babies who had an abnormal ultrasound where there was an additional finding at the level of the chromosomes by doing microarray. So in other words, microarray picks more information that a normal conventional karyotype will have missed. To just go through cell biology so that everything falls into place nicely. First of all, we have a cell and a nucleus, and this nucleus has got 23 pairs of chromosomes. And within these chromosomes, we have the arrangement of nucleotides as they are being shown there. Of course, they are embedded in proteins, but the chromosomes is a double helix made up of the adenosine, a thymine, guanine, and cytosine nucleotides. So they are paired, adenine pairs with thymine, guanine pairs with cytosine. So this arrangement should follow a normal pattern in a human being. Sometimes we could have problems at the level of the bigger chromosome component where there's a total rearrangement of things, or we can have an abnormal number, or we can have an abnormal arrangement of these nucleotides we see there. Cells on their own, normally most cells are in what you call sort of G0 phase or resting phase. Then some cells will be in the G1 phase where they are just growing within themselves. Then cells may go into this DNA synthesis, what we normally call interphase, also followed by cell growth. But there are cells that are always in a state of replication. These are the cells which we call they have high mitotic index, like bone marrow cells, like the cytotrophoblasts we were mentioning. They are always multiplying, and even the fetal cells, they are always dividing because the fetus is growing rapidly. So some cells which are not always dividing, that means for us to analyze them, we might need to stimulate them to divide, to go into the mitosis. The mitosis process is what is being displayed here. In the mitosis process, when the cell starts the division, it multiplies its chromosomes by 2. After multiplying the chromosome by 2, those chromosomes are arranged in the center in what we call metaphase, and they are pulled apart by these spindle fibers so that we have half complement of each. When they divide, they will go on to multiply to return the number of chromosomes to a diploid number, that is 46 or 23 pairs. And then we do have them going again to the resting phase. So for us to study the chromosomes, we need to stimulate the cells to go into the metaphase. The metaphase is that phase which will allow the chromosomes, which are normally coiled together and difficult to analyze. So when the cell starts dividing, when it goes into metaphase, the chromosomes uncoil, unbundle, and they are arranged in such a way that we can analyze them during metaphase. So it is because of this, the understanding of this mitosis phase, and also understanding what happens at the level of the metaphase, that allowed people to study chromosomes in a better way. To just revise a little bit of the cytogenetic nomenclature, the chromosome nomenclature, so that we understand how some of the syndromes are named and so forth. So when you look at the chromosomes, the short arm is called the P arm. The long arm is called the Q arm. So the chromosomes is divided according to these white and black bands. We will describe how these bands come about. So you can see the labeling there from 1, 2, 3, 4. Within these bands, they are further divided again into subregions, as you can see there. And within the subregion, they are further again divided, as you can see. So that when you are labeling a certain region, for example, if you want to label this region, we are going to call this is chromosome number 3 on the short arm, that is the P, then on the region 2 and subregion 2. So this is how the chromosomes are labeled, and this is how they are called in terms of the labeling. Then, of course, to understand that there are different types, there are what you call metacentric. Then we also have the subcentric, depending on where the centromere is. The centromere is that place where the two chromosomes are stuck together. So the centromere can be at the center, we call this metacentric, can be just off the center, this is submetacentric, some call it pericentric. Then they can be closer, sort of closer to the end, that is acrocentric. When they are right at the end, that is called telocentric. So this has also allowed chromosomes to be identified and to be numbered from number 1 to 23. Then we have what we call chromosome bending. What is chromosome bending? Chromosome bending is a process whereby after the chromosomes have been sort of put in metaphase, they are labeled with a dye, or they are mixed with a dye, and the dye will color the chromosomes pattern depending on the adenine, thymine, or guanine cytosine. There are certain stains, like James stain. it will color the adenine thymine region. So all the black stuff you see there is the region that is rich in adenine and thymine. Then we have the C bending, which I didn't put here, but it mainly colors pericentromere heterochromatin. Then we have the Q bending, where quinacrine is used. It's a fluorescent pattern which will be displayed. It is almost the same as the G bending in terms of what it intensifies. Then we have the R bending. The R bending, it intensifies the guanine cytosine bases. So what you see here is the R bending. So that means the R and the black stuff is that part of the chromosome that is rich in the guanine and cytosine. Then we have the T bending, where it's more of the R bends at the telomeres. Why this bending? This bending allows the identification of chromosomes, because when chromosomes are analyzed or are broken after the metaphase and the cells have been bursted, they are all arranged haphazardly. So when they are colored, so this allows them to be identified which chromosomes pair with which one. So this allows the arrangement of chromosomes and also to identify the abnormality of structure at the level of the chromosomes. That's the usefulness of the bending technique. Then let's go to the procedure for the actual cytogenetic analysis. What exactly happens when we take the sample for CVS or when we take the sample for amniocentesis and we send it to the laboratory? So when the sample arrives at the laboratory, they are going to culture it in a medium to allow the cells to divide. This applies to the fibroblasts from the amniocentesis sample and also the mesenchyme from the chorionic villus sampling. Then they incubate for the 2 to 3 days. Within 2 to 3 days, the cells will have divided and most of them will be at the stage of the metaphase. Then they will add what we call the spindle poison. Colchicine is still being used in some centers. Some centers are using other things now, but colchicine, which is a spindle poison, is going to be added to the mixture so that the cells are frozen in metaphase, the stage which I showed you where all the chromosomes were aligned in the center. Then after doing that, the cells are transferred to a test tube and centrifuge to arrive at a concentrate layer. Then when the concentrate layer is obtained, it is transferred to a tube which contains a fixative. Then the cells are put in a microscope, but before they do that, they also add a hydropic solution so that the cells can swell at the base, so that the chromosomes come out of the cells. Then, of course, the chromosomes are then stained, are stained at this level. Then they are identified on the microscope, and photographs are taken, which are called ideograms or karyograms, are taken, and then the chromosomes out of this picture. Before the technology, this used to be done manually. After printing your chromograms, you would manually arrange the chromosomes. You would manually arrange the chromosomes according to the bending pattern to try and decipher the karyotype. It used to be a manual and laborious process, but now all this is now being done by computers. Then the chromosomes are read. Then when the karyotype is done, that procedure is done, what are the abnormalities that we may be able to pick, or what are we looking for at the level of the chromosomes? So we are looking for rearrangements such as deletions. The A, B, C represents sections of the chromosomes. They don't represent nucleotides on my presentation here. So when there's a deletion, that means, for example, you can see this is normal. Then here we do have an E missing there. So that means there is a region of the chromosome that will be missing. So this chromosome may be short if the deletion is very big. Then in terms of invasion, duplication, we have a repeat of a region of the chromosome. As you can see that there is a repeat of the BB there. Then invasion, we have sort of inverted sequence of the region of the chromosome. You can see this is normal, but here it will be inverted. Instead of having an ABC arrangement, it's an ACB. This is all detected by looking at the bending pattern at the level of the chromosome. Then for the translocations we have a situation whereby one chromosome will share or exchange parts of the chromosomes with another. These two chromosomes here, they have exchanged. You can see that the FG has now been put there, and this one is now longer, and this one is shorter. Translocations can be balanced, can be reciprocal, which will create a balance. That means if this one swaps, this one may also give this one an AB there. This is more of swapping, but in certain cases, it may not be like in this case, whereby this one has not gotten anything from the other one, and the translocation can be balanced or unbalanced. That means during the translocation process, no genetic material was lost. The total amount of genetic material is still within the individual. But if some genetic material is lost, then that becomes an unbalanced translocation. Then we have aneuploidy nalisomy, where the whole, all the two diploid chromosomes are missing. And we have monosomy, where one of them is missing. We have trisomy, where there's an additional chromosome, and tetrasomy. And then we have polyploidy, and autoploidy and alloploidy. Autoploidy, that means they are all coming from the same individual, then the alloploid from another species, the extra chromosome component or set. So these are the chromosome abnormalities that we can identify when you do karyotype. To just give you a quick examples, this is trisomy 21. You can see that when the karyogram is analyzed, there are 3 chromosomes. It's chromosome number 21. So this is trisomy 21. Then we see here that when you look at number 13, we can see that these chromosomes are differing in size because there is a deletion that has happened to the region on chromosome 13 there. The region from region 21 to region 33 of the long arm of chromosome 13, there is a deletion there. Then monosomy X is what people normally call tenors. Monosomy X, there will be a missing sex chromosome, whether X or Y, but there is only X there. So there is a missing X chromosome in monosomy X. Then in triploidy, we have a triplication of the chromosomes. Instead of having a diploid number, we now have a triploid number. You can see there are now 3 on each chromosome there instead of the 2. So these are the abnormalities that we can pick using conventional karyotype. What you have to know about karyotype is that it is very good at analyzing the number and the structure, but the structure defect has to have a resolution of at least between 5 to 10 megabase pair. So that this therefore means that if the defect, the deletion, the duplication is probably less than 5 megabase pair, you are not going to be able to pick it up when you do the conventional karyotype, which I've just described. You are going to miss that. This is going to be interpreted as a normal karyotype. In terms of the turnaround time, normally the procedure turnaround time is 8 to 10 days, but in our situation, we normally get our results after 14 days because of the transportation issue to South Africa. Then the CVS sample, if CVS is done, because we said the cytotrophoblast cells are already having a high mitotic index, they already have a lot of cells at the metaphase phase, so it is possible to just quickly spin to poison them and rupture the cells and analyze directly. As a result, preliminary results can be obtained within 24 to 48 hours for CVS by direct analysis of the cytotrophoblast. Then, however, these results are preliminary results. You should always wait for the culture results because of the high risk of placenta-confined mosaicism. Placenta-confined mosaicism occurs in probably, I think, up to 5% of the cases. You may have placenta-confined mosaicism. That means it's just on the placenta, but not there at the level of the fetus. As a result, if you just take result from the direct, you may actually mislabel a fetus as having an abnormal karyotype when it is normal. So we then have to wait for culture, which will take the same number of days as that for amniocentesis. Right, I think on karyotyping, I think I've emphasized that. Then we also have another way of analyzing chromosomes, but this is more of a targeted kind of abnormality. If we know the abnormality and the gene panel or the chromosomal component involved, instead of waiting for the 14 days to receive the results for amniocentesis, we can alternatively choose to do what you call FISH. What is FISH? The FISH is a technique whereby we have fluorescent labeled probes that are targeting certain parts of the chromosomes. So the cells is analyzed in an interface. It is put on a glass slide where it is fluorescent labeled probes have already been fixed on the slide, so that there is hybridization of the chromosomes together with this fluorescent labeled chromosome component. This test is useful for doing what you call targeted analysis for the known abnormalities. What are the commonest reasons for doing it? If we want to just know, is the trisomy 21 there or not? If we want to know, is trisomy 18 there or not? If we want to know if there is microdeletion, 22q11 microdeletion or not, what people used to call DiGeorge syndrome and so all these other syndrome, 22q11 deletion syndrome. So if we want to know these tests, we can order a FISH. A FISH will give us results within 48 hours. In our case, the turnaround time is around 5 days because of the transportation to SA. So we can know very quickly if trisomy 21 is there, trisomy 18 or 13 is there or not. This is how the results are interpreted in the laboratory. To just show you the so this is an example of 22q11 deletion syndrome. So if the genes for 22q11 deletion syndrome, we're going to see the, first of all, the green dye represents chromosome 22. Then the red dye represents the region for the 22q11.2 region. So where the region is there, we are going to see the fluorescent dyes as we see them there. But if it is not there, the dyes will be missing. So that means there is deletion on this part of the 22q11 syndrome. So this is how the results are interpreted for FISH. FISH has got limitations. It's not good enough for a baby who has a defect to order FISH. Why? Because FISH is only going to tell you about trisomy 21, 18, and 13. It's not going to tell you about the other structural abnormalities like deletions in other parts of the chromosomes. It's not going to tell you about duplications. It's not going to tell you about invasions. It's not going to tell you about trisomy 6. It's not going to tell you about these other rare trisomies. So for a baby with a defect, it is not good practice to say you want FISH, which will just target trisomy 21, unless if you are very certain from the clinical features from the ultrasound that, look, trisomy 21 is high on the agenda here. But it is good to study the whole chromosome. Chromosome microarray. when I started the talk, I showed you a comparison of microarray and also conventional karyotype. And in the study which I showed you, it is now proven that chromosome microarray will pick additional chromosomal abnormalities which would have been missed by conventional karyotype, because conventional karyotype is a lower resolution than a microarray. The microarray resolution is between 10 kilobase per to 50 kilobase per, which compares way much better than a 5 to 10 megabase per, which we get with a conventional karyotype. So what is it, this procedure? So what this procedure does is that it compares the test DNA with a test with a reference DNA. The normal DNA will be fluorescently labeled and then put on a slide. Then the DNA from the test individual is also labeled with a different dye, so that when these two are mixed together and they hybrid, so when they hybrid, the intensity that will be generated from the fluorescent after mixing of the colors is then read by a laser scanner. So what the laser scanner is trying to do is to, where there is matching, is obviously going to report a different kind of picture. But where there is variance between the normal DNA and the abnormal DNA, is going to report that as copy number variance. So it's going to report that as copy number variations. This, we see it in duplications. We also see it in deletions. The copy number variants, they are not easy to interpret. There are copy number variants of non-significance. Then there are copy number variants of significance. This is done by genetic people who then have to interpret and say, this copy number variance, is it of significance or non-significance? And it will describe the what is missing or what has been added to the chromosome and outline in terms of how the result appears. It's complicated in terms of how the result appears, and how to read it is very complicated. But the report will clearly state the region on the chromosomes which is either duplicated or deleted, and this is how the process works. So it is this difference in colors that are read by a laser machine, then interpreted as copy number variants of non-significance or significance and so forth. So what does this test do? What is the clinical utility for this test? This test will allow us to pick deletions which are small, which are too small for conventional karyotype, and it will allow us to pick duplications which are too small for a conventional karyotype. Because of its ability, it is now standard practice that every patient who has a fetal defect should not just be done a conventional karyotype alone, but should also be done a chromosome microarray. So, if I go there, so chromosome microarray in other countries, it is now the test that they order. They've actually done away with the conventional karyotype. They just go straight for the chromosome microarray if there is a fetal defect on ultrasound, so that they will take, so that they already take care of those kind of situations where you have a normal karyotype but with an existing major fetal defect. Gene components. So when you look at the gene, the chromosomes, there are lots of regions of the chromosomes which serve different functions. But the most important part of the chromosomes is this part which we call exons, because exons are the ones that code for proteins. The introns are normally the ones that go in between the exons, and we have all these other parts of the chromosomes which are not very clear in terms of their clinical utility in terms of prenatal diagnosis. But because people have realized that this part of the chromosome, which makes up 2% of our genetic material, is the one which is responsible for the phenotype in terms of expressing us, whether we are healthy or disease. It's all coming from the exomes. Of course, it's not 100% true. People are beginning to question, to say, is it true that all these other parts do not contribute to our wellness and our well-being? But it is largely agreed that most of our health, most of our being comes from the 2% of our genetic material. So as a result of that, because still with microarray, we have situations where microarray does not give us enough data after looking at an abnormal baby. So now technology has moved on to say, why can't we just sequence the exons, so that, because the normal exon sequencing has been done through the Human Genome Sequencing Project. So now it is known how a normal sequence of exons should be. So as a part of diagnosing, to see if there's any abnormal arrangement of the nucleotides, that is from the adenine, thymine, guanine, cyanine order, it is now possible with a process we call whole exome sequencing to analyze the arrangement of those nucleotides to see if there's any variation in the way those nucleotides follow each other. This is the process called whole exome sequencing. The laboratory process, of course, involves these are the exons, and the introns are the ones in between the line there. So the process, first of all, involves separating the exons from the introns so that you remain with the exons. Then the exons are hybridized. Then after that, they are sort of multiplied. Then after being multiplied, they are washed, and then they are sequenced. Once they are sequenced, they can now be compared to a reference sequence. And after being compared to the reference sequence, the data will be analyzed this way. This is an example. This is the reference sequence. This is the new exome from the patient. We have samples. So when we sample the patient and we are now sequencing, we can actually see that instead of having a AAA, which is known to be the reference exome sequence for a normal human being, the patient sequence is an AGA. The same here, we can see that instead of having an AGC, we now have an AAC, GTC, and we now have a GCC there. And we can also see here sort of duplication, which is happening at the level of the sequencing. So in other words, what is whole exome sequencing trying to do? The whole exome sequencing is trying to lower the level of diagnosis up to the level of the nucleotides, how they are arranged, the sequencing, to say, has the sequence been disturbed, has the sequence been interrupted? The question now is, what is it that we learn when we sequence the human exomes? But before I go to the human exomes, I want you to know that the whole genome sequencing is now being done. It's not yet being applied in prenatal diagnosis because of the difficulties of interpreting results at the moment. But now there are centers in developed world where you can walk in and you have your whole genome sequence, and you are told your whole genome sequence, and you are told which mutations you are carrying, which disease are you likely to develop, and also your partner can also be told the same sequence, and you can be told what are the risks of you having a baby with a recessive condition and so forth.
after your whole genome has been sequenced. But it has not yet found its use in prenatal diagnosis yet because of the difficulties in counseling patients with some of the results. So, so when we get the result, what information can we get from all this which I've described here? We can identify this non-single nucleotide polymorphism. Each individual may vary in how these sequences are arranged, but in a normal way, what you call a normal variation, whereby instead of you having a GTC there, it can be normal that you may have a slightly different sequence from another person, not in a pathological way. That is called single nucleotide polymorphism, the variation from one individual to another. Then we can also have very rare difference, which may be difficult to interpret and find their meaning. Then we can also see what you call the never been seen difference, which are unique variants. This is a part of the single nuclear polymorphism, but they are not known. They have not been known. You'll be the first person to have them, and people won't know what to interpret, what that will mean to you or to your baby, to the baby who is inside. Then we can pick insertions and deletions. So we can pick insertions. These are the pathological issues that you want to pick to say, is there a missing nucleotide? Is there another nucleotide that has been inserted, as we see here? So these insertions and deletions, they will then be interpreted by geneticists to try and decipher what kind of disease or pathology is responsible for these kind of deletions or insertions that will be seen at the level of how the nucleotides are arranged in our genes. So this is where we are right now in terms of prenatal diagnosis. The whole exome sequencing, what is the clinical utility? How is it being used clinically at the moment? Of course, in research centers, it's now being used more as a core test together with conventional karyotype, microarray, and whole exome sequencing. That means when there is a fetal defect, people are now ordering the whole panel from conventional to microarray and whole exome sequencing for the patient. Then there are other centers that are saying they've already moved over now. All they are now doing is whole exome sequencing. They are no longer, they don't want to waste their time doing the conventional karyotype. They just order straight away the whole exome sequencing from the word go. Then what is the most pragmatic way which we can use it? I was talking to the Lancet Lab manager to find out how far, whether they accept samples for whole exome sequencing or not. I'm here to get a response. But what is the pragmatic way to use this? Probably the whole exome sequencing can be used as a contingent to the conventional and microarray. That is those babies who have an obvious defect or a multiple defect or a syndromic kind of defect, and you have done both the conventional karyotype and the microarray, and they are all normal. Probably that would be more of a wiser way of using the whole exome sequencing. The cost is still exorbitant. I think it's still costing more than the microarray at the moment. The microarray in some centers cost between 500 and 1,000 at the moment. I think this is my last slide on the whole exome sequencing. So I just want to do a quick recap before I finish to say prenatal diagnostic tests have migrated a lot from the conventional karyotype so that it's no longer good standard practice to just order conventional karyotype in the presence of a fetal defect. It is good to at least do a chromosome microarray so that we don't find ourselves in this bizarre situation of telling a woman who has a baby with a defect that your baby is a normal karyotype, when in actual fact, there could be a defect that the conventional karyotype could not pick. We should also know that the whole exome sequencing is now here to answer some of the questions that were left hanging by both doing the conventional karyotype and also the microarray. Thank you so much. You can follow us on our social media pages. Our presentations are posted on our YouTube, Fetal Medicine Foundation Zimbabwe, so you can follow us on this social media site where we post our presentation. Thank you so much. I'll leave the room to the chair, the time to the chair. Thank you so much for your time.
Thank you, Dr. Verenga, for that very informative talk, which I'm sure will help us to triage our patients who we see having obvious syndromic appearance on scan. Please feel free. I'm going to open now the floor for anyone with questions, comments, suggestions. Please feel free to contribute.
Morning, Dr. Klachaya.
Morning, Dr. Ndagu.
Yes, I have a question.
Please go ahead.
Hello. Okay. Thank you, Dr. Verenga, for the detailed presentation. I have a question on conventional karyotype for a sample that was collected using chorionic villus sampling. You said that we can get preliminary results in about 48 hours, and then they can go ahead to process the specimen further with a culture, and then to give us another result in about 10 to 14 days. So how does the culture rule out the placental mosaicism, seeing that we are using the same cells from the placenta?
Thank you, Dr. Ndagura, for the question. Remember, we're not using the same cells. Like I said, when I was doing a recap, that the chorionic villus sampling, there are two sets of cells in the chorionic villus sampling sample. We have the cytotrophoblast and the mesenchyme.
Okay.
So the cytotrophoblast have a high mitotic index. They are always dividing, right? Because of the high mitotic index, they are prone to division errors, to mutations, and all those things.
Okay.
So because of that, they may not represent the actual cells, the actual mesenchyme. Even the mesenchyme can still have what you call placenta-confined mosaicism, whereby we will need to do an amniocentesis to break the tie. But there are a lot of divisional errors that are subjected at the level of the cytotrophoblast because of the high mitotic index in those cells. That's the reason. That whatever abnormality you pick should always be confirmed by culture. If there is mosaicism at the level of the mesenchyme, then amniocentesis has to be done to make sure that the mosaicism that you saw is also on the fetal fetal fibroblasts, which are in the amniotic fluid.
Okay, okay, that's fine. And the other question is, so if we collect a specimen for using amniocentesis or CVS today, we are going to order a microarray. Which diagnostic test are we going to order?
Both conventional karyotype and microarray.
That's okay. Okay, thank you so much.
Dr. Ndagura, this applies to a baby with a fetal defect, remember. But if the woman has got a high risk just from screening and the NT is below the 95th centile, we just order a conventional karyotype. So this applies to a fetus who has got a nuchal translucence above the 99th centile, that is above the 3.5 millimeters mark, and a baby with a fetal defect. We have to order both a conventional karyotype and a chromosome microarray. But if the woman comes to us and she screened a high risk from the combined test or from fetal self-free DNA test, and we check the baby is absolutely normal, and the number of percent was within normal range or less than 99 percentile. In that case, we just order a conventional karyotype.
Okay, thank you very much.
Welcome.
Dr. Verenga, is the conventional karyotype the one which is costing about $500 at Lancet?
Yes.
Okay, when we add microarray, how much then does it cost?
It's going to cost more, probably 1,000, to be honest.
Okay, that's quite
It's going
steep
to
for
cost more.
population.
Yeah, it's going to cost more. Unfortunately, it's going to cost more. This is one of the reasons why we need to increase the screening rate. Was the situation which is happening. One of the reasons why we are doing these talks is to conscientize every obstetrician and every probably general practitioner out there that the only way the laboratories will be able to do local processing or even to do a karyotype, we need to increase the screening rate to offer as many women as possible prenatal screening for aneuploidy, so that we can increase the invasive testing rate, because our invasive testing rate is very low at the moment. I think at best probably we may do 1 or 2 amniocentesis in 1 to 2 weeks. So because of this, I don't know other people who are doing amniocentesis, how often they are doing them. But if the laboratory is going to be culturing 2 amniocentesis in 2 weeks, it may be overly expensive for the laboratory to do that. But if we increase, let's say, our invasive testing rate, whereby probably nationally, if we can maybe have 10 to 20 karyotypes per week, I think we can convince the laboratory to say, look, it makes business sense not to send these samples away. Instead, they would rather do them here. So we want obstetricians and also the GPs who see patients first to offer women prenatal screening for Down syndrome, Edwards and Patau, and all the other aneuploidy, so that we can have a group who are high risk, who need the invasive tests. Because at the moment, we only see fetal defects who are being diagnosed late and so forth and so forth. And yeah.
There was a question which had been posted by Dr. Chamunonga asking why we are still sending samples for karyotyping to South Africa. So I think that answers Dr. Chamunonga. I'm not sure, Dr. Chamunonga, if you want to make a follow-up or if you have fully answered.
I think I've been answered. Thank you, Dr. Hloche.
Okay, thank you. Anyone else with a question?
Probably just let all the participants know that we also do amniocentesis in the public sector. Of course, the patient, right at the moment, there's no fee structure, so they're not paying anything for the amniocentesis in the public sector. However, they still have to take their samples to Lancet for cytogenetic analysis.
And for sorry, Dr. Verenga, please go ahead.
I was saying for CPD points certificates, don't forget to send your email address and your full name to FMFZim so that you can be sent your CPD certificate for attending the webinar.
Thank you, Dr. Verenga. Please note to all other participants who joined us for the first time, this is a recurring meeting which we have every Friday from 8 to 9 a.m. Please feel free to join us. If there are no other questions or any additions, suggestions, then I think we'll close the meeting. Thank you all.